Morning, everybody. I'm Shyam. I work as a junior research associate at the Center for Predictive Human Model Systems at Atal Incubation Centre, CCMB. So, welcome all to the sixth webinar of the series in developing human relevant research in India. So, for the sixth webinar, we have two industry leaders in vitro science. One is Dr. Subhadra Dravida, the CEO of Transtoxbio Biologics, and Dr. Subramaniam Vangala, the CEO of Reagene Biosciences. So before we start, I would like to introduce Dr. Surat Parvatam, the Senior Research Associate at CPHMS, and she has done her PhD from TIFR Bombay and also is a prolific science writer interested in engaging with people who are involved in human relevant research. So I invite Dr. Surat to start his presentation and to speak about CPHMS for a couple of minutes. Over to you, Dr. Surat.
Thank you, Shyam. I'll just take 2-3 minutes of your time just to briefly talk about the Center for Predictive Human Model Systems, which is organizing this webinar series. Just 1 second, I'll just share my screen.
Just a small request, kindly keep your audio off. Meeting.
Okay. Is the presentation visible?
Yes, yes.
Blue heart. Okay, I am from, like Shyam mentioned, I am from, Shyam, can you mute everyone? Okay. Yeah, so like Shyam mentioned, I'm from Center for Predictive Human Model Systems, which is a center which was established at Atal Incubation Centre, CCMB, in collaboration with Humane Society International, India in 2019. And this center was primarily established to enable the human relevant research in India and to understand what are the challenges to conduct this research in India and how we can further advance these methodologies. So to do this, I'll just briefly talk about in 1 or 2 minutes what are the various activities that we are doing. So few of the activities that we have written a few white papers on various these human relevant methodologies such as organoids, organ-on-chips, integrating frameworks such as adverse outcome pathways, and tools to develop predictive models for human physiology, which are like systems and computational biology. So I've written 3 white papers on what are the challenges to do this and what is the state of funding, regulation, infrastructure in these fields. So this was primarily done to understand what is further needed to advance these fields. And apart from this, we also carry out several educational and outreach activities such as workshops. And this webinar is also an example of that, where we conduct a monthly human relevant webinar series where we have people from mostly Indian researchers who come and talk about the research that they're doing in how they're developing or using these human relevant model systems. And we've also recently given out a grant to build an adverse outcome pathway in the field of cancer, and it has been currently awarded to 2 Indian scientists who are developing that. And in the similar vein of increasing, because one of the major limitations that we found is people are not aware of what are the non-animal and human relevant methodologies which are based on human cells or human biology in India. So to do this, we also, apart from conducting webinars, we've also written several popular science articles and reviews to again increase us in this area. So with this short introduction, I would stop and ask Shyam to introduce our first speaker. And yeah, thank you, Shyam. Shyam, are you there?
Yeah. Thank you, Dr. Surat. So moving ahead, we have the first speaker, Dr. Subhadra Dravida, who is the CEO of Transcell Biologics. So Subhadra Dravida leads the team and collaborations responsible for discovering druggable formulations across therapeutic areas and platforms. Uh, she also requires in converging and executing both laboratory clinical research programs and other such aspects. So in the past, she has been affiliated with the University of North Carolina and the Ottawa Health Research Institute, USA. And also she has been associated with CCMB in Hyderabad in the Eye Research Foundation. Her expertise lies in the adult stem cell technologies, the predictive platform development and screening, primary cells, low throughput and high throughput platform technologies, cancer progenitor cells, repository of of patient source screening platforms, cell-based readouts, drug discovery, and pre-discovery process. So having said this, I would now request Dr. Subhadra to take over. Dr. Subhadra, over to you. Uh, Dr. Subhadra, just a second. Dr. Subhadra, yeah.
Yes.
Now you're muted. Yeah.
Yeah. Can you see the screen? Yeah.
Yes.
Okay. Thank you, the organizers, for this nice opportunity to connect with the participants. Yes, I'm Subhadra, and in this opportunity, the presentation, I would be touching upon human surrogate systems, few human surrogate systems and their adoption in proposed adoption in industry. Yes, like all the researchers, including basic researchers to the industrial researchers, we all must have used some of these preclinical platforms as models in our discovery and developmental pursuits. So to name a few of these well-established and then thoroughly utilized models happen to be, of course, in silico, keeping in silico aside, yeast-based, zebrafish-based, and of course the very popular Drosophila-based models, and some saline-based as well, including animal models and all. These have been very popular in understanding, like, the test materials, behavior, and then the features, while the intent and then the motive has always been to mimic a human condition outside the human body. So with that intention, like, many of these platforms have been very widely used and utilized in preclinical research stages. So that includes, as I said, like, discovery and developmental cycles. Now, I think billions and billions have also gone into coming up with the best model and then, like, suiting the requirement. But then somewhere we sincerely believed that, like, the relevance to human application has been lost or kind of compromised or maybe diluted. And then on top of it, the cruelty that is caused to the animals, and then in using these animal-based models for developing, like, some kind of a solution to humans has been like, on the radar for I think for several years, like, till date. So that is the the genesis of any mindset kind of thinking in terms of coming up with some alternatives to the existing and then deep rooted platforms and models in preclinical research. And not just the pharmaceutical industry and then the cosmetic testings, but also like, you will be really surprised to know that this kind of an alternative thinking process has even reached the vaccinology segment also. So because now that, like, we are kind of all have survived this COVID attack, I'm sure, like, the talk and the discussion is all about the vaccines that are being developed for COVID. Even in for research of developing vaccine candidates and then manufacturing, the interest has always been to have humane endpoints in evaluating the vaccine potency, along with developing some replacement models to the primates and then, like, the monkeys, especially if one has to test the neurovirulence caused by some of these vaccine candidates or vaccines that are being manufactured. So I'm just kind of bringing the relevance of pharma and biopharma industry, at the same time, like, the compelling need for some advanced and then, like, human surrogate in vitro systems to be adopted in pharma and biopharma industry. So while there is this compelling need, at the same time, like, the parallel research and then interest has always been looking at some of these stem cell based models, because, like, they kind of exhibit very interesting features, like, that could be nicely capsuled as benefits and advantages attributed to the models that are developed utilizing these stem cells as platforms. Yeah, so that is the context. And also I just want to kind of connect with you all that, like, of course, not the researchers, like, somebody who is not part of the scientific community, although, like, these are all the different models that have been practiced in preclinical research stages. It is not like we have seen some evolution and then, like, as of now we don't use those yeast based or, like, zebrafish based or Drosophila based models. Like, we have adopted different models for different contexts. So we still, majority of the basic researchers, do still work on Drosophila based models, and then even sometimes zebrafish models also. So although, like, we have been thinking that we have to work on certain human relevant models and platforms, we have always been forced, the scientific and industrial community is always forced to pick something which is already kind of published and then, pick some clues from the existing literature. So we kind of got caught into our own vicious circle, like, where we have to use the existing platforms and then, like, develop on the existing systems. So but then, like, that has been the pain point, while COVID has actually, like, dragged all of us onto the streets and then, like, taught us that we have to think out of the box and then, like, work on the revolutionary models, like, that have relevance to human application, and it's not that like, it's not this particular human surrogate systems have not been in existence. They have always been there, like, different groups have been working on it. It is just the gap in terms of embracing and then adopting to the context is what is has been missing. So Transtoxbio, Transcell developed Transtoxbio has all new league of human-sourced cell and tissue-based in vitro systems for the user, enabling him or her to understand the pharma and biopharma assets better and faster. And we also believe that like this particular vertical-based platforms have the power to empower clinical trial prognosis. Now, allow me to, answer this question. What are these human surrogate in vitro systems? These could be alternatives to few existing systems and could be complementary models to few other existing systems. And these are cell and tissue based configured in vitro platforms, while the composition of these platforms dictate the use cases. So I'm only referring to Transtoxbio residing platforms, and I'm defining like, what are these and then the characteristics of these platforms in the context of use cases. So if what has been answered, then like, why should anybody be even looking at these human surrogate systems is like, for relating to human research, because the source of these platforms is human. And when I say human, like, it could be a healthy donor or could be a patient also. And when it comes to modeling, building any complex model on top of this on top of these systems, they're very meaningful in terms of yielding specific readouts that actually require no extrapolation. So for aeons, like, the research has always been towards extrapolating the readouts to human species, but then by integrating and adopting these human systems, the user need not extrapolate, and then like, it predicts and then is related to the human specific application that the user is working on or like, developing towards. So these are some of the reasons why one should be even looking at adopting these human surrogate systems. Now, these systems are developed to be phenotypically responsive, and one can evaluate the reactions, like, the genotypic expression acquired from these platforms. At the same time, these platforms are functionally approachable also. So all in all, like, the human surrogate systems have this excellent proven capability to predict the behavior of the test material in clinics. Yeah. Now, to talk about some of the features of these platforms, they are definitely in vitro and real time. So when I say real time, I want to differentiate them from the in silico platforms that are also kind of being practiced in drug discovery and development. And one of the highlighted features is that the cell and tissue components are harvested from biological discards, human biological discards. So that is the unique selling proposition over here compared to the other cell lines or like animal sourced or biopsy sourced cells and cellular components that are already kind of being practiced in research and developmental activity. And all of these platforms are primary in nature. So primary in the sense like, they're not transformed. Yeah. And progenitor, human based. So progenitor is like, in fact, it captures all the stem cell specific features into one platform that is configured. So these are some of the interesting attributes to the researcher to build on top of it. And then because the source is available in abundance, there is this ability for large-scale production of these platforms that any researcher would definitely fall in love, like, to exploit this particular advantage, utilizing this the platform. And then in terms of obtaining homogeneity of either the cellular structures or like the tissue that is being utilized, that is also given from these platforms, and they can be prepared as free of bioburden. So this is also an important characteristic feature of this platform. If one has to compare with some of the biopsies like skin biopsies, where they're full of bioburden, and then it is impossible to obtain the sterilized formats of those kind of platforms. So by employing these human surrogate platforms, the researcher can obtain reproducible, sensitive, and specific test readouts. And these platforms are amenable to different kinds of methods and methodologies to be built on. And all of these platforms, like, have been tested to be suitable for established logistics, and they can be stored at low temperatures till the user intends to get the working solution from the stocks, and then prepare for the intended experimental use. And they are like, one would be probably very curious to know, like, are these platforms, the utility of these platforms specific to only cancer research, or like, for any kind of a specific disease condition? No, at least Transtoxbio residing platforms are disease agnostic, because the technology that we have developed in configuring these human surrogate platforms, like, sources both from the donor as well as, like, the disease bodily biological discards. And another important advantage is, like, in terms of the ethics part, because we don't deal with any biopsies or, like, any kind of the embryonic source. All the sources that we have been utilizing in developing these configured platforms are ethically immune to harvest the required components. And in the labs, they definitely mimic the human physiology. So there is this 100% relevance to the human milieu. And of course the composition is, like, adult progenitor, either cellular or tissue components. And I don't need to probably differentiate, like, the uses of primary components, primary cells versus, like, immortal cell lines, because the learned audience and then the participants for sure, like, must have experienced even the differences, between handling primary injury cells and then the transformed cell lines. And then, like, when there is this opportunity to handle progenitors, one can, the experiment can have the luxury to enjoy, some of the differentiators in terms of the microenvironment that the experiment is required to, recreate, and then mimicking human physiology, and then to certain extent genome copies also. So the Transtoxbio based human surrogate in vitro systems enjoy and then exhibit all of these features, like being primary in nature, and then composed of progenitor components, and then are not immortalized, presenting to the user in terms of the differentiators. Yeah. So see, in drug discovery stage, there are certain critical endpoints, like evaluating the attributes in terms of the test material to be exhibiting. These human surrogate in vitro systems, like are a big hit, because the researcher would get the endpoints that the test readouts that the user is actually wanting to see, because each stage endpoints, like would dictate the fate of the next stage. So what I mean to say is, at the initial chemical library screening stage, if the parameters are so critical, and then they are specific to human relevance, it makes very convenient for the experiment to go to the next stage, which is evaluating some of these hit molecules. Yes, in fact, I remember very well that I was touching upon this particular property of transdifferentiation. So some of our platforms exhibit this potential to transdifferentiate upon induction to multiple lineage-specific cellular systems. So this is a very, very important and a novel a property to do with the pluripotent stem cell types that can be, I want to go to the next slide, that can be exploited when it comes to the drug repurposing domain. For example, drug repurposing is all about finding a new function of the known drug. So there is a great scope for our human surrogate in vitro systems to participate in the hunt and quest for finding new functions of the known drugs. So the best example here I just want to cite is the big company, Big Pharma, Novo Nordisk, is big time into screening and hunting for the chemical libraries, and then some of these drug candidates having the ability to generate beta pancreatic cells on stem cell based models. So that actually becomes the starting point for the research to be directed and focused towards the disease condition that the thrust can be put onto in terms of both the efforts as well as the resources and then investments. So but for this ability to transdifferentiate, to the best of our knowledge, there are no other in vitro systems that can undertake this kind of the ability to participate in drug repurposing. So it is the composition at the same time, the responsiveness of these in vitro systems that make them qualified to participate in different stages of drug discovery and development. Yeah, Sham, can I go to the next slide, please? Yeah, and then during drug development, especially if it is a cancer drug development program, then again, the patient-sourced tumor-derived organoids, or tumorous spear-based platforms, which are again human surrogate in vitro systems, can as well find interesting participation to support the developmental readouts. So it's again specifically in selecting some of the drug candidates, and then pruning them to become the investigational drug candidates for cancer medicine. And then if it is in vitro ADME, that is what is adopted by the industry. Again, the human surrogate in vitro systems can find their role and spot to participate, if the readouts are in establishing cardiotoxicity kind of safety related endpoints. Because again, if the platform is of induced pluripotent stem cell type and composed of those particular subtype of cellular components, then they have this ability to become cardiocytes that can become the platforms for validating and evaluating the drug candidates exhibited any cardiotoxicity. Next slide, please. Yeah, and leaving that pharmaceutical research, if one has to look into the cosmetics testings domain, there are certain fundamental and basic tests like microbiological testing, and then evaluating the biocompatibility of the cosmetic products that are developed, and then looking at some of the tests like skin permeability validations, and then in vitro residual antibacterial tests, and all, for all these basic tests, the industry need not at all go to the animal models, and very well can nicely settle down to just adopting these human surrogate in vitro systems as completely replacing any other model that is being practiced. So on top of it, some of the other tests like evaluating any exhibited carcinogenicity or mutagenicity kind of validations, these human surrogate in vitro systems are the best fit to participate, and then provide the readouts that are human relevant, for which these either cosmetics or consumer goods are being developed or kind of marketed for application. To the next slide, please. Yeah, in vaccines R&D and production, there are 3 identified assays that become, both the safety specific and potency specific validations, like endotoxin assay, or the cytokine storm assay, and the other one is neurovirulence test. So for these 3 tests, we believe, and then we have some in-house data generated also in terms of the compatibility and the adoption of human surrogate in vitro systems to perform those validations on the test candidates. So other than these, there is no other best or anything closer that the industry should be even looking at using as platforms to incorporate in their existing processes and tests. Next slide, please. Yeah, and for some of the immuno-oncology programs, humanizing mice models is again, like, a very well established methodology where the mice body are utilized as bioreactors carrying functional human genes, cells, or tissues or organs. So this is not new, but then, like, the adoption is already kind of seen in the industry. So this as well stands out as the best example where the human surrogate in vitro systems have already found their spot in the research and developmental activity relating to the human condition or the application. Yeah. So next slide, please. Yeah. So to summarize, I just want to kind of give some, the vantage position point of view in adopting these human surrogate in vitro systems. Like the source has to be definitely human, and then again the human biological discards should be the option, not the biopsies, like, where for some of the access biopsy cannot be the source at all, and then, like, the researcher would be left high and dry in terms of the reproducibility, and then establishing specificity and sensitivity, like, if at all the platforms have already kind of been utilized. And then the platform has to be definitely primary in nature, and then composed of progenitors, nothing else. Then sometimes if it is induced pluripotent platform composition, it is the best, not necessarily always. So keeping these as fundamental features to be constant, any methodology built on top of it in terms of modeling, yielding, like, the human specific endpoints are best suited to interpret in terms of their usability. So it's actually a new league of the platforms, next generation in nature, and then, like, revolutionary for the industry to quickly adopt and then make their processes compatible as well, like, to establish their usability. So we also kind of believe that, like, it is not the regulator's headache in terms of advising or, like, making it mandatory to the industry to choose what kind of platforms they should be using. It is to the user, like, who is the industry, to decide in terms of the options, while the regulatory agencies would only be interested, like, to see the data, ultimate data that is established on the test material. So it's definitely going forward, like, all hail only human surrogate in vitro systems, either directly into adopting or, like, using them as platforms to model further in the industrial research, translating the applications to human benefit. Yeah. So, okay. Yeah, that I think I'm done with my presentation, and happy to take any questions if at all.
Our next speaker is Dr. Subramaniam Wangala. So he's a very experienced pharma scientist and an executive with more than 2 decades of leadership experience at global pharma in the USA. He has worked with companies such as 𝐒𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬𝐬 the CROs in India with SciLife and Advinus. He brings with him a unique insight into providing cost-effective alternative solutions for substituting animal studies for regulatory products. He has received his PhD in biochemistry from the Memorial University, Canada, and a postdoctoral degree from University of Colorado at Boulder. and another postdoc from University of California at Berkeley. He has a vast and immense experience in the pharma and drug industry, and also has 50 plus publications in peer-reviewed journals and several invited book chapters. And currently, he's serving as the CEO of Reagene Biosciences, based in Bangalore. So we are glad to have Dr. Mani with us today. Over to you, Dr. Mani.
Thank you. Can you guys hear me? Some people are saying that they can't hear very clearly. Just wanted to double check. Everybody can hear, right?
Yes, Dr. Mani.
Okay.
Yeah.
Yeah. So the bandwagon right now is going on for alternative to animals. There's a great research tools. I mean, in fact, with the last 40 years, nearly from my PhD days, I started using in vitro systems along with the in vivo systems as well. And in academia, I used a lot of in vitro systems along with the animal models. Whereas in the pharma industry, I also had a chance to really look into the clinical outcomes, where what do you predict out of the in vitro systems? Do you actually see a successful clinical outcome or not? I'm not really focusing right now on alternatives to animals to clinical outcomes. I'm more focusing right now in this particular talk is about how the in vitro systems of human origin predict the human outcomes. I think that is the most critical question we need to ask. And the one reason I'm saying that because the humans are so diverse. Basically, they're genetically different. They have lots of differences, gender, variabilities, age, height, sex, all kinds of things do matter in terms of predicting an in vitro system to have a clinical outcome in humans. So the predicting from in vitro systems from human to so in vitro human surrogate systems to human outcomes is a critical thing, and I'll present you a few case studies on that that I worked on a couple of, and of course some of them are also from other researchers. So the next slide, how do I go to the next slide?
Dr. Mani, do you want to switch to the presentation mode?
Where is that?
It's the below, the next button next to that. Yeah, this one.
Okay, is that okay now?
Yes. Yeah, and then you can push the down arrow whenever you want to move to the next slide.
Okay, yeah. So I'll give you an outline of PBPK models, introduction, and what are the clinical and population variables? What are the variables that actually impact our outcomes or predictive values of these models? Available PBPK models and applications to date. What is the in vitro versus in vivo correlation? I'll talk about one subject from environmental pollutants is benzene. And the case studies too is about curcumin. You know, this is a health ingredient. People look at it a lot, showing that it may be anti-cancer drug or anti-arthritic drug or something. People have a lot of faith in curcumin. And also I'll present one case study about the pharma drug I worked with and the in vitro versus in vivo clinical outcomes. And I'll finally wrap it up with how do you build 3D models for PBPK modeling and future prospects, okay? So a basic PBPK model has two components. For example, orally, if you give a drug, it goes to the gut, which has a stomach and intestine, everything. And the drug which is absorbed goes through the portal vein into the liver. And that is the primary entrance. And from liver, it gets distributed to the rest of the organs. Whereas if you give an IV dose, it's distributed differently. It goes through the venous blood directly, goes through the lung, and then gets distributed through the arterial blood into the different tissues. So the pharmacokinetics, if you look at the barriers of oral drug versus the IV drug, may be totally different. So when you do an in vitro assays and trying to put this into models, you have a lot of panel of in vitro assays. I think probably for one drug, I may have to use about 25 to 30, whether they're pharmacodynamic models or pharmacokinetic models. So you have about 25 to 30 models that we typically use before we actually say, hey, this drug is safe enough, correlating with animals as well as for the humans, coming up with a human first-in dose, saying that, okay, this is the actual dose that we need to give it to humans, and this dose is going to be the safe dose, and it also be efficacious dose. And in the same time, with these PBPK models, we also compute, is it going to be a narrow therapeutic drug or a drug with a wide therapeutic margin? So those are the things that we'll look at before we actually hand off to the clinicians and say, hey, this is a very good drug, you can take it further. But the problem starts when you go into the clinical trials, phase 1 clinical trials. 95% of the drugs that are handed over to clinicians never make it to market. Okay, I'm not just talking about animal models, I'm also talking about we have extensively used in vitro human surrogate models as well for the last 25 years or so. And I'll try to show, because this is too complex a thing, that you're also trying to look at the pathways, building AOP models, and looking at the model output with in vitro versus in vivo correlation, and you're building a computational model with all this data that you have in front of you, okay? So in vitro to in vivo extrapolation is the critical thing. And what are the components in this PBPK model? There are extrinsic factors, there are intrinsic factors. If you look at also drug-dependent factors. So if you look at on the right side corner of the slide, drug-dependent components that typically include ADME factors, pharmacokinetics, pharmacodynamics, and mechanism of action. In that, there are subsections like metabolism, active transport, passive diffusion, protein binding, drug-drug interactions, and receptor binding, etc. If you go to the left side of the thing, there are lots of extrinsic and intrinsic factors. So drug-drug interactions, of course, is again, because another drug comes, that's an extrinsic factor. And you also have intrinsic factors like age, race, organ dysfunction, disease, pregnancy, lactation, gender, genetics, others. So all these things also matter in terms of the environment, medical practice, regulatory, other, all interfere. Then there are other components like system components, drug independent. Whereas is it rapidly perfused organ, for example, lung versus the kidney versus the liver, or slowly perfused organs. So the blood is flowing through all these organs, and how the drug is transported through these organs matters. I mean, in terms of not just transported, what is the blood flow rate, and are the drugs are being taken up or metabolized in only one organ like liver, or metabolized in different organs. So you bring all this data together, whether in vitro data or in vivo data together, and you predict and learn, and again you confirm that in human outcome is positive. When human outcome is positive, not positive, that is prospectively you fail the model, so you come back with the retrospective model again, and so it's a kind of iterations of models that you keep doing it several times. So here are several variables that you can actually look into. Gastric pH, gastric emptying time. Of course, solubility is one of the major components. Many drugs are actually today that are made by pharma industry are not soluble, and so they actually have very, very poor absorption through the gastrointestinal tract. And the pH is another matter. If it is with too acidic pH, many drugs actually get degraded right in the stomach itself. And gastric emptying time, site of intestinal absorption, and gut transporters and gut metabolism, especially the kinetics part of it, liver transport and metabolism, enzyme kinetics is very, very clear. And then there are routes of excretion, biliary excretion versus urinary excretion. So then also all these things also matter in terms of the disease state. If somebody is impaired with kidney function, then the drug may have to be treated differently for that patient. Same thing with hepatic impairment, because if there's a hepatic impairment, the drug metabolism, drug transport status is different from the normal person. Ethnicity, we have known that ethnicity is a major driver in population pharmacokinetics, because if you go to Africans, if you go to Europeans, if you go to Indians and Asians, the genetic polymorphisms of these genes of drug metabolism and drug transport are huge, and they do affect in terms of the population differences and toxicity efficacy. So apart from that, of course, drug-drug interactions, especially people above 55, geriatric patients, they're not taking one drug, they're taking 5 to 6 drugs, maybe because of different complications they have. So they interact with each other, and that can modify their metabolism, transport, and can be toxic many times. In fact, cardiac toxicities do occur due to the drug-drug interactions. So you can link all these things in your model. Look at it. I'm not going to go through the entire slide here. I just want to show you that how do you predict an intrinsic clearance of a chemical in PBPK model. The red box you can see at the bottom is intrinsic clearance. That intrinsic clearance can vary with a lot of different things that I've described, all these variables that. So distribution in populations are very different for a lot of these things. Okay, just one example here taken from Freddie Boy et al. Paper. They showed that with age, isopropanol clearance is actually changing quite a lot. So basically what's happening is that age has an effect in drug disposition. So if you say that, okay, my drug is, this is the dose for the normal 18 to 50 years old people, then you have to really estimate what is the dose going to be for aged patients separately. Okay. And this is a graphical representation of a population toxicokinetic model, and where you actually have, so if you look at in that model, that you can see the square boxes like E, which means exposure, T, which means time. But the circle with theta has a lot of unknown variables, and you're linking those unknown variables to the exposure with the time and trying to come up with that Y, which is the actual data, and trying to come up with some factors, trying to build a model. And this is how you build the models for population pharmacokinetics. So you can see that exposure graph is given on the top, which actually that variance of the exposure, sigma square, you can find out what is the variance in the population total. Okay, so environmental pollutants, typically people have used retrospective and epidemiological data combined with mechanistic models. Typically, back in 1990s, people have used Monte Carlo simulations and Bayesian simulations, etc. And when you come to drug discovery predictive models, mostly these are prospective models now, and uses both in vitro data plus animal data combined. And there are several more models. If you see CAT, which is transformed to ACAT and ADAM recently, which is basically a physicochemical tool. But these physicochemical tools are now integrated in another platform called GastroPlus, which is an oral absorption tool.
Dr. Mani, you muted yourself. There is a button called mute, which is next to your video. So I think that has.
Can you hear me now?
Gotten.
Yes, yes, yes.
Okay.
Yeah.
Somehow it got muted. I don't know why.
Yes, we can hear.
Going back to the slide again, as I said, environmental pollutants, people from 80s and 90s have typically used Monte Carlo simulations and Bayesian simulations derived from the epidemiological data, because most of the environmental pollutants, toxicity and cancer data obtained through the epidemiological data, how many people have become sick, etc. Using that data, and you build the in vitro tools as well as the animal models and get mechanistic data, and with the mechanistic data you try to model the PBPK model. Whereas in drug discovery, these are more predictive and prospective tools. Prospective means you don't even know what is happening and you're trying to predict something that you don't know. So in vitro data plus animal data is combined and use this for prospective modeling. And there are several models. There are physicochemical models that now have become outdated and now got incorporated with more advanced models like GastroPlus, which is basically an oral absorption tool, which is a proprietary and commercial tool. SIMSIP is another very popular tool because of the cytochrome P450, which means CYP enzymes are simulated because we have so much data now with the human CYPs, and they have very good physiological models to predict the human outcomes with the metabolism data. And you have to choose the right model that is relevant for your case, rather than just using these models randomly and without having prior any knowledge about these models. Okay, next one. Oh, okay. What happened? I'm still not able to move the slides.
Dr. Yeah.
Okay. Just want to make sure that, okay. So, as I said, drug metabolism. Drug metabolism, if you look at, there are at least 4 different cytochrome P450 family of enzymes, which belong to cytochrome P450 1A, 2C, 2D, and 3A have been implicated in drug metabolism in the liver primarily. That is called phase 1 metabolism. And CYP3A is the one metabolizes 50% of the known drugs, and CYP2D is the ranks number 2. But in any case, the phase 1 metabolites may undergo drug-drug interactions with other drugs. If the same enzyme metabolizes 2 different drugs, and that they actually can also lead to immunotoxicity, cardiac arrhythmias, because one drug inhibiting another drug metabolism, the other drug concentrations go way up above the toxicology threshold and cause cardiac arrhythmias. And the drug may also have mitochondrial damage, DNA intercalation, exaggerated pharmacological effects. And these are all, drug itself can do it, sometimes metabolites do it too. So the phase 1 metabolism can also result in reactive oxidative stress. This oxidative stress can lead to damage to DNA and protein, which is called oxidative DNA damage. And most of them are reactive metabolites, either electrophiles or free radicals. And then you have phase 2 pathway, phase 2 metabolism, which is primarily conjugation with the glucuronides and other pathways and detoxification mechanism. This metabolism actually leads to nonlinear pharmacokinetics in most cases. So if you don't know the metabolism, you are trying to do pharmacokinetics, you have no clue whether the drug is going to be, pharmacokinetics is going to be linear or nonlinear. So you need to know thoroughly what is the drug metabolism and drug transport for these drugs. For example, as I already mentioned, CYP4 families are there, right? You can look at the table top table. CYP3 is about 50% of CYP3A4 primarily. 50% of the known drugs are metabolized. If you take pharmacopoeia, there are about maybe 3,000 drugs there, out of which 50% are actually metabolized by CYP3A4. And CYP2D6 about 30%, and the rest of them are actually smaller, lower. But CYP2C enzymes also play about 10% role. And if you look at the liver, human liver, the variability between the humans, if you look at 2D6, that's about 1,004 variability in one ethnic group, not just different ethnic groups. If you take 1,000 people and look at their 2D6 levels, they're quite variable. So everybody will metabolize differently with the CYP2D6. Same thing with CYP3A4. It's not as, widely described, but CYP3A4 is only 90-fold different, complete. So basically what's happening is that you have all these enzymes that are very, very differently expressed in the human liver. So you humans are very unique in terms of having in vitro directly correlating with the in vivo situations. In many cases, if somebody is has some disease, and especially immune disease, they produce cytokines, and the cytokines actually inhibit these enzymes too. So you don't have these enzymes if people have some disease states like diabetes, arthritis, and cancer, etc. So you need to be really understanding whatever data you generate in healthy volunteers may not be applicable to the disease state. Okay. And there are a lot of transporters now. I mean, I'm not going to go into this transporters, but again, there are drug transporters in hepatocytes, blood-brain barrier, intestinal epithelia, and kidney. These are all the major sites the drug goes through. A drug first comes through the intestinal epithelia if it is an oral drug, and then goes into the liver where biliary excretion is also a major part instead of the drug going into the systemic circulation. Then blood-brain barrier, if you're trying to develop a drug for neurodegenerative disorders, you need to have a drug which has good blood-brain barrier. Poor blood-brain barrier so that it can penetrate better. And the kidney proximal tubule. Kidney is an excretory organ, so you also need to know what are the enzymes that can modify your pharmacokinetics. So I'll talk about a little bit about benzene. I've worked a lot on benzene as an academic scientist back in 80s and 90s. In fact, majority of my PhD thesis on how benzene causes leukemia. And it's still continuing. And I'll tell you the end story that it's not easy to do these models. So the epidemiological data indicates it's a human leukemogen, causes leukemia in humans, particularly acute myelogenous leukemia. The occupational exposures are very, very low chronic doses. I mean, it can be 0.05 ppm or something. And we have done extensive in vitro and in vivo mechanistic data. I probably spent a lot of time understanding the mechanisms. And the benzene is Ames negative. Benzene is Ames negative, but it induces chromosomal aberrations, so particularly numerical chromosomal aberrations. It's highly metabolized in liver, but actually the secondary metabolism by myeloperoxidase in bone marrow is the result why the benzene causes leukemia. So it's not just liver metabolism that is important here. You also need secondary metabolism in the bone marrow by myeloperoxidase, which is critical to form the oxidative stress. So we have created extensive PBPK models and employed in risk assessment to determine the occupational exposures of limits. So I'm not going into detail here. This is how the metabolic pathways are charted out. And also you can see the DNA damage by reactive oxygen species as well as by the reactive electrophiles and causing the mutagenesis and clastogenesis. We also had to determine the enzyme kinetic parameters like Km and Vmax to understand the specificity of each metabolite. It's like not the drug-drug interactions. Metabolites are actually interacting with the parent drug. So when you have 5 metabolites interact with each other also with the enzymes. So this is the pathways that we have worked a lot. And you can see that 8-oxo-G, that is 8-hydroxydeoxyguanosine, which is a reactive oxygen species damaged DNA, that is a biomarker. So you can actually quantitate that and see how much damage is there by oxidative DNA damage. And here it is, we looked at bone marrow of mice, and you can see benzene at 200 milligram per kilogram gives you highest damage as you go increase, maybe because the increased doses probably are not metabolized and inhibiting each other's metabolism. And you also look at the metabolites, right? Benzene itself, maybe leukemogen, but when you look at the phenol, hydroquinone, catechol, those metabolites really didn't do much of oxidative DNA damage compared to the control mice. But if we combined phenol plus hydroquinone, phenol plus catechol, and etc., these combinations actually gave more oxidative DNA damage. So these metabolites interact with each other in a toxicokinetic manner. They get dumped into the bone marrow in more readily, and more concentrations of these metabolites in bone marrow results in myelotoxicity or leukemia. So we also looked at in vitro system like human HL60 cells. There are 2 metabolites, benzene triol and hydroquinone we used. You can see that there's highest oxidative DNA damage at 30 minutes, But surprisingly, it's coming down to 60 by 60 minutes. Maybe because DNA repair is very extensive. So DNA repair is another parameter that is a confounding factor that we need to know whether the mutations are coming from DNA repair or direct damage by the drug itself or chemical itself. So I had a friend and who was a colleague actually at Berkeley, Frederick Bois, and he took all my data. I mean, very, very good mathematician and statistician, excellent capability. And he started building these models in 1992 with the benzene. And he created that benzene model with our data at that time. And then you can see that, in fact, he has created more and more evolved models as the more and more data is coming through in vitro and in vivo data, that he has created more and more models. And if you look at the last one, in November 2020, one second, next one. So here it is. Current status of benzene, limit of occupational exposures and risk assessment. This is published in November 2020, just a few months ago. So when after 1992, the first physiologically based pharmacokinetic model was deployed, we're still trying to understand, trying to see what is the risk of the benzene concentration. So these guys have alluded that lowest hematotoxicity potential is at 2 parts per million in the air, with 8 hours time weighted average per day. And of course, no effects were seen at 0.59 ppm. Same thing for genotoxicity also, they've seen very low levels. Finally, with the conservative approach, regulators always want to be conservative. So they said should be 0.25 ppm to ensure that people, whoever gets exposed to beyond 0.25 ppm has to be very, very careful. And so 8-hour time weight average is now proposed very recently. So this is an evolving situation. Every time you think you know something, then there's some other parameter that you don't know that comes. And all these things are actually put into the AOP models as well. AOP has been part of the benzene assessment as well. So now I'll talk about another one, curcumin. Curcumin, everybody thinks that it's a great antioxidant and it cures cancers, any other diseases, etc. And it's a great herb. That's what. But the perplexing problem is that it's poorly bioavailable. If you give curcumin, it's not even, you can't detect any curcumin in the blood. So that's a major issue with understanding how curcumin works in the body. And the major reason for that is curcumin is actually extensively metabolized in the intestinal wall to a glucuronide metabolite. So the correlations between antioxidant activity, beneficial effects were debated because of the curcumin is poorly absorbed through the intestine. Here is the structures of the curcumin and it's a glucuronide metabolite. So if you build a model and you take the plasma concentrations of these drugs and trying to correlate the epigenetic and the antioxidant activities to the drug. So those are the biomarkers that you look at to see, does this, even in the absence of curcumin in the body, you can see these epigenetic markers and antioxidant activities to see if you have efficacy of the drug. So they did an observed and predicted blood concentrations of curcumin and its glucuronide metabolite. This was recently published by a group. If you look at the bottom chart, plasma concentration of the curcumin is below 0.1 nanogram per ml. In fact, it's not detectable. And they made a simulation with little bit data that they have. But basically, curcumin, you cannot see it in the... And some people claim that if you give curcumin with piperine or fenugreek, it may be absorbed better, but we don't know. I mean, there's not enough data on that. And so they actually modeled the COG, glucuronide plasma concentration, based on the population variability of the age, gender, sex, and everything, all kinds of, even UGT enzymes. And you can see observed versus predicted levels are quite good here in this case. the triangle, red triangle is observed, and the predicted is the just solid line that is going through in the middle, okay? And they did a curcumin capsules they gave and look at the blood for the curcumin glucuronide plasma concentrations, looked at the PKPD modeling, and looked at some of these antioxidant levels in the leukocytes, or white blood cells for PD. So here are all the pharmacokinetic parameters for the curcumin glucuronide that they derived, is observed. And in fact, UGT, UDP glucuronyl transferase, involved in the metabolism. There are 4 at least, and all these things were used in the SIMCIP, the physiological model I described earlier. And that curcumin is also metabolized to some extent in cytochrome P450-1A2 as well. This is the PBPK. I'm not going to go into the details. This is all simulation models based on the one data you have from a few subjects. You're looking at the variabilities in the population. And the same thing they did for the epigenetic and antioxidant measures. You can see that there's a significant change in these parameters, showing that curcumin does have these gene expression changes, suggesting that it has some biological activity. Despite curcumin, you don't see it in the circulation. This is how you do the PKPD, PBPK modeling. The last one I'm talking about, a case study 3, it's a compound X. I was working with a compound which is already gone into the clinic. We've done a lot of work, in vitro work, in vivo work, and using human surrogate models as well, in animal models as well. We predicted this is going to be the pharmacokinetics of the drug even after you dose it for 2 weeks for the drug. And here's the data that I'll show you, that very interestingly, that the left chart, if you give the drug daily for 12 days, right, at 3 different doses, 5 milligram, 10 milligram, and 20 milligram, you can see the at 10 milligram, it's more than dose proportionally high, the drug concentration. This is not linear anymore, which was very surprising to us, because we thought single dose should be able to predict the PK of even the multiple dose. But something happened here, we don't know what happened. And we actually looked at the metabolite also. And if you look at the metabolite AUC, although it was there at the highest dose at 20 milligrams, but you don't see that at 5 milligram and 30 milligram, 10 milligram doses. So there's something happening to the metabolism of the molecule itself. So we looked at some predictive model here. There's a model called CYP3A inactivation model, because cytochrome P450-3A4 metabolizes this drug. So does it inactivate this enzyme so that can explain our higher parent drug concentration? So the metabolism is inactivated. And in fact, there's a model where you actually, cytochrome P450-3A4 has a synthetic versus degradation kinetics. So it itself gets synthesized and it gets degraded. And you have to put that synthetic degradation kinetic cycle into the apparent inactivation by the drug model. And we looked at the AUC inhibited by the dose over 12 days, and the degradation is also, enzyme degradation is also put into it. And to cut the story short, so this kinetic model we applied, if you look at this 3 or 4 degradation rate is 0.00083 minutes in over 14 hours. And
Dr. Mani, sorry to interrupt. Yeah, I could. You have like 5 or 10 minutes more, because it's very interesting talk. But yeah, for the interest of time.
Sure.
Yeah, thank you.
So for 100 milligram dosing, we have predicted plasma levels of 97 nanogram per ml, which is 0.14 micromolar. And we also looked at the plasma protein binding and see what is the free drug concentration. Based on that, we said that cytochrome P450-3A4 can be inhibited by 62% just with single dose. So if you are giving multiple dose, then you have entire enzyme. So it's actually overwhelming even for synthesis and degradation cycle of the cytochrome P53A4 itself. So we have predicted AUC about 2.6-fold increase, but we observed a fold increase about 10-fold, greater than 10-fold. So even the cytochrome P53 inactivation kinetics is not predicting for the 10-fold increase in the AUC over 12 days. So we looked at something else. There is another missing link here. In the intestine, there is a P-glycoprotein, which is the actual drug transporter. And in fact, this P-glycoprotein can inhibit some of the drug getting into the systemic circulation. Now, the drug itself is also inhibiting the P-glycoprotein. So, the concerted action of the P-glycoprotein inhibition and CYP3A4 inactivation, both are contributing to the 10-fold excess. And this is the model that we published also. In fact, we looked at the backup molecules in the program. There are other chemicals, thinking that maybe other chemicals would be better molecules than this. But we looked at it, and all of them are chemically similar, and also had the same problem when we did the in vitro models. And the entire program was terminated because of the chemical series that is going to be forming reactive metabolites that actually inactivate the CYP3A4 and P-glycoprotein. So this comes to another point, is that many drugs that reach the market show some idiosyncratic events. You don't see these problems in the animal models, right? So if you look at the person withdrawn, hepatotoxicity, about 25, cardiotoxicity, about 16. So you have a lot of these drugs are withdrawn. If you look at over the last 50 plus years, there are more than 200 molecules which went into the market, and then FDA had asked to pull them out because people are dying because of these drugs, either through drug-drug interactions or idiosyncratic immune toxicity or kidney toxicity or cancer potential for these drugs. So PBPK applications in drug discovery should be focused on predicting the FIH safe dose, number one. Number two is the drug-drug interaction and dose determination in special patient groups, pediatrics, geriatrics, or pregnant women, or even disease patients. So you have a lot of these models need to be refined to be able to also incorporate the changes in blood flow, drug metabolism, disease variables, pharmacogenetics in special populations, etc. So I'm coming back to the how do you build an in vitro model, right? The problem, I've used purified enzymes, subcellular fractions in 1980s, and as moved into 1990s, I used 2D cells, tissues, recombinant enzymes, and in 2000s, 3D cells, tissues, etc., regenerative stem cells. Now we also have something called microfluidics that is emerging as a powerful tool. The problem is there are some advantages and disadvantages. If you look at in 2000s when we used cells alone in the 3D platform like hepatocytes, we still don't have cell-cell communication, multiple target identification in pharmacology and toxicology. The problem is that, as I said, a drug may be metabolized in the liver, but it's also some of the metabolites go to the bone marrow, and they are the ones that are toxic, not the parent drug itself is toxic. So you need to have a model that actually incorporates all these things. Some people are using tissue slices, putting into the microfluidics. And I tell them, why are you doing that? We are putting the whole liver into it. But you don't have blood circulation because blood goes into the liver through veins and arteries. If you bathe the liver tissue with buffer and blood, it's not going to do anything for you. So you need to have a circulatory system along with these models. It's not just putting a cell and looking at it. A liver has 5 different cell types, so they all communicate with each other. And that is very important, in fact, for the communications, so this 3D models, I think, is the next level that we are looking at, microfluidic enabled models. And this is a little short story. I tried to work on this model back in J&J. And there was an article also published, and I was already, at that time, these devices were coming up, and we applied through the Hurel model. And in fact, we generated a little data on that, and scientists actually, new scientists in that, a freelance writer, acknowledged this contribution. Basically what we did was we created a prototype. We used adipose, liver, and lung with some drugs, and we're looking at how do you flow. I mean, this is a flow system. You input the drug into input changer, like a drug goes into the oral mouth and going out to the kidney, but we don't have a kidney here, but we're trying to look at combining multiple tissues, what would happen. So this is the hepatocytes, human hepatocytes. We've taken it on the static model, like you put it on the matrigel coated plates and looked at the enzyme activities. And we are the same hepatocytes. We looked in the microfluidic flow device with the lung tissue and other tissues are enabled. And you can see that the drug metabolism enzyme activities are 10 to 20-fold higher, which suggests that you need a flow system. Static cell models people are using today, even today, and those models really don't predict what happens in the in vivo because there's cell-cell communication and organ-to-organ communication. Just think about it. Hormones are there, right? Hormones can stimulate these enzymes too. So endocrines systems. So you have a lot of other things that are happening in the in vivo system that are not able to mimic these in vitro static cell models. I'm not going to go into we also did 10 compounds for the toxicity issues, and we are showing in this model, just to tell you that we were able to predict the liver toxicity better with the microfluidic model than the static cell model. Okay, that's what the bottom line for this slide.
Dr. Mani, we won't have time for questions, so if we maybe we can...
Okay, I'm almost done. This is the FDA submissions. They're increasing. A lot of people are now incorporating the FDA's PBPK models into the FDA submissions. And the FDA also has a guidance document. And also peer-reviewed papers are also increasing quite a lot recently. So the take-home message, PBPK models require extensive in vitro data from in vitro human surrogate tools. Each model is case-specific. And the existing in vitro human surrogate tools are time-consuming, and you need 25 to 30 models to look at, and they do lack cell-cell communication, organ-to-organ communication. So consolidation of in vitro tools into a 3D flow system can help build robust PBPK and PKPD models and enhance the in vitro in vivo correlation for positive clinical outcomes in humans. So that's the bottom line, that you need to have a 3D bioprinted microfluidic models to be able to mimic the human out of human. Thank you.
Thanks a lot, Dr. Mani, for the very interesting talk. So maybe we'll just take 2 questions and then move on to the first speaker. So, I'll just take one question from Sesha Sai, who's asking what is the difference between permeability rate limited and perfusion rate limited models?
Sorry, again, say it again. I could not hear the question.
There's a difference between someone's...
There's a lot of noise coming, background noise.
Yeah. One second, I'll just mute everyone.
Dr. Pankaj, but yeah, go ahead.
Yeah, so the question is, what is the difference between permeability rate limited and perfusion rate limited models?
Yeah, so the perfusion is very important in terms of keeping all the drug transporters and drug metabolism intact. So if it's a poorly perfused liver, then it's not going to have the optimal activity because the signal transduction and all these events that are happening that impact the gene expression of the hepatocytes can be limited if there's not enough perfusion of the blood through these organs. So it has to be highly perfused, number one. Permeability is more different from the physical chemical parameters. If it is a fat soluble molecule, it may not penetrate the membranes very easily. That is the reason many of these lipophilic molecules don't have good bioavailability, although they're very good efficacy-wise in vitro and toxicity-wise in vitro you can predict, but you cannot. So permeability also is dependent on transporters. There are specific transporters that regulate. These are all gatekeepers, basically, hey, Gurkhas, and saying that you cannot get in. That's it. If that Gurkha says that you cannot get in, you cannot go in. So these drugs also have Gurkhas preventing them going in. It's a evolutionary system that biology adopted to ensure that you don't get toxic molecules into your body. Okay, that's
right. And second quick question, which Lindsay is asking, she's saying, Dr. Mani, you make very good points about huge variability of human population in vitro models can help predict some of this, but do we need to rethink clinical trials to address this? Phase 1 would seem to be very unprecedented.
So, I mean, most of the prospective models now are so far so good. I mean, they do predict better. The problem is there are 30 models out there. And this is where, I mean, I have to put a lot of money and spend 2 years using these 30 models to generate the data to predict what happens in the clinical trial. So I need to shorten it. And so I need a PBPK model, which is with a 3D model where I can combine all these 30 assays into one, or maybe two at the most. And so I can save a lot of time, and the predictive accuracy is going to be much better with these models than the single standalone in vitro models. And so your PBPK model is more actual human mimicking model here with a 3D model than with standalone 30 models that you generate. Okay?
Thanks a lot, Dr. Mani. Thank you, Dr. Subhadra, for great presentation. So again, I think we'll just have time for 1 or 2 questions. So I think there was a question in the beginning when you had given the first time. So I think there was a question that how do we study systemic effects using these in vitro models? I think there was a question. So
just to give it that vaccines are systemic in their biochemical activity. So how will we using these particular cell lines give us an overall picture regarding the action and side effects of a particular vaccine or drug in an organism?
Right. So again, like, you know, a word of caution for some of these applications, at least I'm not pushing or representing that, like, you know, you can completely replace the existing systems that are being practiced. But for few where, like, you know, there is this scope, for example, like, you know, neurovirulence, and the other one is cytokine storm assays, and then the third one is endotoxin validations. For such plain vanilla kind of methodologies and then the tests, you know, the integration of human surrogate in vitro systems can be seen. So those are the low hanging, in my language, like, you know, where the industry can adopt. Not like the suggestion is not to completely replace and then go about only looking at these platforms. Yeah.
Right. Which I think most of the others, they are mostly comments. Yeah, I think rest are more like comments rather than questions. So if there are no other questions, then we can thank Dr.
Excuse me, I want to ask question.
Sure.
Should I ask?
Sure.
Go ahead.
Myself, Dr. Pankaj Kumar Singh from IIT BHU. The lecture was very nice. Both of the lectures I have listened. That is nice. One simple question is that in concern with the PK, that is the pharmacokinetics and the PD, pharmacodynamics, I think both of these, the 3D model is you have shown. So how these models can help in the study of or in the development of new vaccine? Can you explain, both of you or any one of the both the person? Thank you.
Yeah, thank you for the question. Dr. Mani, would you like to go first?
Yeah, so you're looking at efficacy. See, look at take for SARS-CoV right now, the virus that we're all working with. If you understand the biochemistry, the major interaction is with the ACE2 protein, and also the aiding protein is TMPRSS2. These two are very important in lung cells, epithelial cells, that will actually internalize the SARS coronavirus inside. Now, if you know that the ACE2 receptor is the binding, and if you understand the biochemistry of the ACE2 receptor binding domain, and you can actually create either drugs that inhibit the ACE2 receptor or the antibodies that you generate against the spike protein should be able to inhibit the ACE2 receptor. So you can evaluate in a lung, we have created a lung vascular model in our company, and we are actually using that to screen drugs for the efficacy through ACE2 receptor inhibition, or we're also looking at antibodies that can inhibit these ACE2 receptors. So you could do evaluations like that. Based on that, you can actually select the right molecules to go forward into the drug repurposing way, or the antibody therapeutics that you want to use. So I hope I answered your question.
Yeah.
Okay. This is the matter of the development. Okay, I understand. But what about the clinical trial? Whether it can help
Actually, even in India now, there is basically because immunology, animals versus humans is totally different, completely different. So animal models are not preferred much because you really can. Although people have done with the monkeys, especially for the Pfizer, BioNTech, and everything, but the problem is. Monkeys are not humans, and many cases, although they're saying that in monkeys they can prevent infection with the vaccine, but in humans, right now they were saying that about 12,000 Israelis got infected even after vaccine was done. So animal models are not really foolproof. The human trials are very foolproof, and which means that you need to have alternative models that can actually predict the viral neutralization through the antibodies and vaccines, and that has to be done. And right now, because I think this year we are on pandemic and hurry, a lot of things have been cut short to get the vaccines out. But we'll learn more and more because these viruses are also mutating so fast, and is the mutating virus has the same affinity for the ACE2 receptor or different affinity for the ACE2 receptor, or has another protein that it actually uses to internalize itself. So we are still working in a vacuum. We really don't have much information on the virus itself. Okay.
Okay, thank you, sir.
Dr. Subhadra, you like to add anything to that?
I think Mani did address.
Okay, yeah.
So thank you both the speakers for very interesting talk. So and thank you for all the participants for joining it. But before we end, I would also like to just briefly request you all to fill a survey. So I'll just share my screen. So this is a survey that the Center for Predictive Human Model Systems is doing in collaboration with with several collaborating partners such as the Italian NGO OSA, the European Commission, Technical University of Denmark, a Euro Group of Animals, and so this survey is basically being conducted to understand more about the animal-derived materials and reagents in scientific experiments. So as we all know, one of the most essential parameters for any biological experiment is to ensure data reproducibility and reliability. And to do this, there is like even OECD has recommended that we need to completely understand understand the knowledge on what are the various sources of the sources and more information about the materials and reagents that are used in the experiments. So this survey is basically being conducted to understand what are the animal-derived materials and reagents that are commonly used in vitro experiments, and what are the main issues that are perceived during the use of those reagents, and also what is the current knowledge of the non-animal derived alternative materials and reagents in vitro experiments. So we'll share the link of the survey in our chat box, and I would request you all, whoever is involved in using any kind of model system, whether it be in vitro, in vivo experiments, if they're involved in, I would encourage them to fill in this survey, and also maybe circulate to other people whom they might be knowing of, who for whom this survey would be of interest. So with that, I'll just thank you for joining in, and we'll just put the link in the okay, yeah, Sham has already put the link in the survey. So I would request you all to circulate the survey as wide as possible, and also fill in the survey yourself. So yeah,
and,
Sham, do you want to add anything?
And I would just like to thank the speakers who have taken their time out and given these interesting talk on in vitro human surrogate systems. And we also apologize for the glitch which happened, for the technical glitch. And if possible, I would request everyone to switch on their video for a minute so that we can have a photo together, if that's okay. So I would request everyone to whoever is part of the meeting to just switch on their video so that we can have a photo for our memories. And also, if Dr. Madhu, if you'd like to speak something.
I'm fine. I think
in the meanwhile, yeah,
I've been following the whole talk. It's great to hear all these possibilities and all that, and also we're very happy to look at the participants, and I'm sure these things will ignite some people to get into this activity and then add more value to this particular thing. Yeah, great. I mean, Surat, it is good initiative. Thank you very much, and also the speakers for having taken time out. Dr. Dravida and then Bengala, thank you very much for your time. It's a pleasure to have you around. Thank you very much, and we look forward for your counsel in the future too.
Thank you, Madhu.
Yeah. Thank you all for your kind participation in the webinar, and hope to see you all in the next installment of our webinar. And also, please, as everyone said, the link for the EU survey is in the chat box. So if any one of the participants are actively engaging in vitro, in vivo, or any related kind of activities, so they are free to fill the survey, and also if possible, kindly circulate them amongst your peers. So having said that, I again thank every one of the participants as well as the speakers who have taken their time and attended this webinar. Thank you all, and see you in the coming webinars. Thank you.
Don't forget about the pictures.
Yeah, sure, sir. I've already taken it, but yeah, for another thing, another one.
Yeah. Okay.
Yeah.
Thank you.
Thank you all.